Hello, welcome back to another episode of Let's Talk Climate Change Conversations. It's a great pleasure to have with me Professor Matt Vijayan from the University of Calgary, and he's visiting us to talk to the students about his work on environmental physiology, environmental stress, and toxicology. So Professor Vijayan has a very interesting career because he did an undergraduate degree in agricultural sciences from the Tamil Nadu University of Agricultural Sciences in 1982, and then in 1984 masters from the University of Agricultural Sciences here in Bangalore, and then went to the University of Guelph for a PhD. And now looks at environmental stress, toxicology, using fish as a model system. And I think the results that the work that you're having is a lot of implications for us in India. He's received multiple awards for his work, published over 200 papers, and it's going to be, I think, a great pleasure and a joy to talk to you today.
Thank you. It's my pleasure being here, and thank you for having me.
Same.
It's such a beautiful campus here.
So I'll start with a question we often start with, which is where did you grow up and how much was your early childhood shaped by the environment?
Interesting question. I grew up I was born in Kerala, but then I grew up in Chennai. So this was a long time ago when everything was nice and quiet and not very crowded. So I was close to the ocean when I was growing up, and did interact with a lot of fish people at that time. So that sort of got me interested in fish at the early age, but that was really not my sort of career choice. Initially, I wanted to go into medicine, and I was unsuccessful. I wrote all my exams and everything, so I got high marks, but I wasn't unable to get into the medical school. So then I got into a professional program that was agriculture sciences. So it was sort of serendipitous as to how I got into fisheries, because I got into agriculture school and then I got into this fisheries program. So that's what started my sort of career. Was decided by what field I got into. But I always liked animals. I loved animals when I was growing up, and so that was sort of a career path I would have chosen even then. Either going into medicine or doing something related to animals.
You have pets or an aquarium?
Oh yeah. We had aquarium at home, and my grandparents had pets. We had dogs and cats. It's a whole family of cats and dogs over there.
Okay.
So yeah, we're always surrounded by animals, especially in Kerala. Yeah. I can't talk about like a defining moment that the career path I have right now was sort of decided by me unable to get into med school when I was in India, and then getting into this professional program that I got really hooked on to. The actual career, I was always interested in sort of biotechnology. So when I first wanted to move to Canada, I still remember writing to people in Canada saying I want to do a program in biotechnology. And it so happened my uncle was in Canada at that time, and he went and spoke to a professor at the University of Guelph, and he said, I'm not a biotechnologist, I'm an endocrinologist. If he's interested, he could come and do a master's with me and then switch to a biotechnology program later. So that's sort of how I moved to Canada. And that was a big move, because when I finished my program here at Agriculture Sciences here in Bangalore, I got a faculty position. Yeah, because we were the second batch from Tuticorin to finish fisheries science. So they were starting to take students after masters into, they were giving positions for few people, and it so happened I got it. And it was a big movement at that time, whether to take that or go for higher studies. And my dad was vehement. He said, you should, follow your career path, and you seem interested in, studies overseas. So maybe that's what you should pursue. So he was really instrumental in me moving, because at that time getting a faculty position in India was a big deal.
Exactly.
Yeah. So he sort of helped me in making that decision and making my move to Canada a lot easier. Yeah, so this is a long time ago.
It was very interesting, because I think many people talk about getting into professional courses. It's changing in India a little bit.
Is it? Okay.
But it's still the idea that if you don't get into a professional course, and then your life is not made.
No.
I think it's interesting, because obviously you really love what you do.
Yeah, absolutely. And at that time I was sort of disappointed when I didn't get into medical school. I could have tried again, but I got sort of upset, that I didn't get in. And so when this opportunity arose going into agriculture school, I didn't think twice. I just took it up and moved on with it. And then I also had chance to go back into medical school when I was in Canada, which I decided not to. But funny thing is now my research is all biomedically oriented.
That's sort of
So yeah, so I took it.
interesting.
It's a complete circle.
Not humans, but it's fish.
No, but I use fish as a model. So
to relate.
yeah. So all my the application of my work could be for
Could be-
biomedical.
Right.
Absolutely, yeah.
So how much of this do you think you got preparation for in your undergraduate or master's degrees? Master's was also fisheries.
Yeah, masters I did in aquaculture.
Okay.
Yeah, so that was very very targeted fisheries oriented.
Applied in a very narrow.
Yeah,
Okay.
but I really liked physiology when I was, I took some physiology courses, both in my undergrad and in my master's. And I really got hooked onto physiology. At that point in time, I thought, if I'm going to do a PhD, that's what I'm going to be doing, either physiology or molecular biology.
What did you like about physiology?
What I like most is, how things move or how things happen, how things work. But not just seeing, you do something and something happens. It's more like what makes it do that. So that's what I think I was always been interested in the mechanisms, right? And even my research now is very mechanistically oriented. And yeah, so just seeing something change really doesn't excite me unless I know what caused that to change. What was the mechanism that allowed something to change or something to grow or something to reproduce. So that's sort of the approach I took in my research as well.
Because I just to give you a little bit of, I did an undergraduate degree and we had a triple major in Bangalore University, and that was microbiology, chemistry and zoology.
Yeah.
And then I moved to the Indian Institute of Science, which had then the first integrated PhD programs in biological sciences.
Okay.
And so that was in 89 to 92 was my BSc, and 92 I moved to IISc. And the first 2 years were a master's in biological sciences, of which 6 of the 7 departments was some form of molecular biology, developmental biology, microbiology, molecular biophysics. Ecology was just the 7th department.
Oh, I see. Okay.
And I just completely by serendipity, I had landed up listening to a Professor Matt Vijayan, who was very well known ecologist in India, and deciding to join him for a PhD. But I was the opposite in that sense. I loved reading about molecular biology. I found it but I found the lab very boring.
Yeah.
So it sort of really depends. And that's why I was asking you, why do we like, why does something sort of...
And sometimes it comes with experience. Like I had no exposure to any of these things until I went to Canada. So it was all very theoretical when I was... and the fishery science program was all focused on anything to do with fish. You can name fish, you can identify fish. They teach you all about, capturing fish and going fishing and all sorts of things. But the physiology was sort of interesting because there was one professor, he was in Canada, he did his postdoc in Canada, and then he came back and he was teaching, and that really got me interested. So he was talking about how you look at fish movement, how you can measure how fish move, again from a functional standpoint, and that really caught my attention. And so somewhere down there, I really like physiology. And even if I had gone into medicine, that's probably what I would have specialized in, endocrinology, physiology. But then when I went to Canada, this professor, Professor Leatherland, he said, you can, my lab is doing stress research and looking at corticosteroids. And he said, you can do a master's and then you can move on to another lab. He gave me several names and said, you can go do your biotechnology program. Because at that time, biotechnology was just starting to grow.
Exactly.
Yeah. And there was...
So this would be the early 90s?
Yeah. Or even mid-80s. Mid-80s, yeah. So it was just starting and especially in the fish field, they were starting to develop, fast-growing fish and all this. It's just starting off. And so I got really excited with that area of, using genetics to improve stock and all that sort of stuff. But then when I came to Guelph, as I was starting to do my master's, I realized I really like what I'm doing here because it was physiology, it was endocrinology, and it was stress physiology. So I went and told John that, I think I'm going to continue my PhD here, and I don't want to. I can do biotechnology for my postdoc. Yeah, so that's how this all happened.
Okay.
Yeah.
Right. So, what did you do for your PhD?
My PhD was looking at how stress affects fish growth, and at the physiological level, we're looking at hormones. Specifically, the question was corticosteroids. That's a hormone we all have. Fish also have, and that's the beauty of fish. They have everything that we have, and system-wise, and so we were using fish as a model to ask the question: if they undergo stress, how is that affecting the growth? What are the pathways that are being affected, and which hormones are involved in that in those changes? So that was sort of the question. So I got introduced to endocrinology, how you measure hormones, and he was very particular about research methodology, and that always stuck with me. like, you have proper controls, make sure, repeat your experiment, make sure what you're seeing is real. So that really stuck in my mind, and I really enjoyed that whole aspect of coming up with a hypothesis, testing it, making sure, you ask the questions correctly, and you plan experiments accordingly to address those questions. Yeah, so that's essentially what I did for my PhD. Was looking at how stress affects growth and what are the roles hormones play in facilitating that.
Right.
Yeah.
And what do you think about mentorship then? I mean, that you got,
Yeah.
let's say, in India or didn't get in India, and in your PhD and postdoctoral work.
Yeah.
What has stood out to you about the kind of mentors that you got?
There was mentors in India for sure, but I think at that time when I was there was restrictions in the facilities and what could be done in India at that time. And but certainly there were mentors, who guided me and said, and there was few of them who encouraged me to go overseas. They knew I was really interested in studies, and so they actually quite encouraged me to go overseas. They said, like there were a couple of people who said, don't take the faculty position, you can always get much better positions later, but because once you stop your studies, you're not going to go back to it.
That is, yeah.
Yeah, so they played a very prominent role in my decision making for sure.
And no, but overall, if you just think of, I mean, because now I've also saw on your CV that you've mentored over 50 masters and PhD students, which is a lot in a career.
Oh yeah.
So what do you think are the kinds of lessons that you learned from mentors that has influenced the way in which you mentor students?
Oh, that's a good question.
And because there are many young people watching this, so
Absolutely,
what should people look for in a mentor?
People should look for someone who can you can talk to first and foremost, and I think the mentor should be someone who can listen to what others are saying, and then not just force their opinion on people, and listen to them, and because different people have different needs. Especially when I'm mentoring, I've mentored so many people, and some come, some are so prepared, some are not. So you have to change according to what the needs are of the students. And I think a good mentor is someone who does that. And I think who listens to people and who guides them and encourages them. I think that's really, really important. You need to give them the confidence. And some people naturally have the confidence, some don't. And so they need a lot of, talking to and explaining things and all that. But I think eventually it really shows. The happiness I get is when they graduate and they go on to do something great, and it really makes me feel really, really good.
Exactly. One of the things I was thinking back to my own mentors is the freedom that they gave you to choose something. I mean, I think you had a lot of freedom, but also at the same time the care of rigor.
Yeah.
You don't want to be left. It's that balance, right?
Yeah.
I mean, you were talking about rigor, something that you learned from your PhD supervisor. So how to do experiments, especially with experimental and field-based research.
Absolutely. Yeah. So what I do is, so the approach I take is my students, who I hope they don't listen to this, but when if they come as a master's student or PhD, the first 6 months I tell them, you decide what you want to do. You take that 6 months. It's sort of like a learning curve. You come, you think about things, what you would like to do. Try something, it may fail. It doesn't matter. You try it. But for master's student, I say I give you 6 months because you have only it's a 2-year program. So within that 6 months, if you can't come up with something, then I'm going to give you a project, and you do that project. But for PhD, that's they have to they have to come up with something they want and that they are interested in, and most of them do. And like I said, some people have the background, some people don't, but it doesn't matter. You give them the time and the flexibility, they always come up with really interesting questions.
And do students come from all across the world?
Yeah. I had students from India. I had a lot of Canadians. I had students from the US. So I do have students quite a few.
And when you have students from outside, let's say North America, do they then take projects that are relevant to their context? So is this typically Canadian?
Interesting question. If it's a PhD project, usually it revolves around the research program that I have. So that it's a broad umbrella. So if I'll tell them, anything related to stress in fish is open game. You can or stress in animals. So anything biomedically related, but stress related. It could be mental health. It could be growth. Yeah. So it's very broad. So then I give them, I give them the opportunity to go and figure out what they want to do. And the reason for that, this is what I really need to appreciate, Canadian funding system. It's called NSERC, Natural Science and Engineering Research Council. The funding they give is sort of for curiosity research. So yeah, so we are not, we are not accountable for what we proposed. As long as the research gets done, it's an interesting research, and we are training people. So that freedom that funding agency gives me really allows me to open up to other students, and so they are given the flexibility to choose whatever they want, as long as it's a solid project, and it fits within this broad umbrella of stress and fish and the role of corticosteroids.
And then I was just thinking of this because yesterday I was working with other colleagues on the idea for an 8-year grant.
Yeah.
And it's very hard, no? We're sitting here and saying in year 8, this is what I want to do.
Yeah.
The world will change, problems will change. You actually should not be specifying what you want to do in year 8, because if you specify it and rigidly stick to it, you'll have a bad project.
Absolutely.
And you just have to wonder who was thinking what when they designed these kind of things.
Yeah.
But this is very nice.
It's a
Curiosity
beautiful
research.
program. Yeah. So they give you funding for 5 years, and it's highly competitive. But for the 5 years, they give you the money, and then they'll ask you what you're doing. And then after 5 years, when you go for the renewal, they look at your productivity. They look how many students you train, what your program is, what you're proposing. And so the future funding is based on that. And it's all curiosity research. It can be applied if you want, but they really don't care, as long as it's good science and you're training people.
So can you give me some examples of the wide range of things that people have done? Because you said there is quite a range.
Oh, in my lab?
In your lab.
Yeah. So I have 2 streams. So that's the NSERC Discovery Grant is all curiosity driven, but then you can also apply for applied grants, which are more, of biomedical relevance or environmental relevance. So yeah, so I have in my lab I have 2 streams. One is curiosity driven research. Half of my lab people do that, and the other half does environmental work. So they're looking at contaminants in the water, and how that affects animals, and it could be anybody, even humans. And some of these grants are being funded by the local municipality. City of Calgary has been a funder for my program. They've been supporting my program quite a bit. So what we are trying to figure out there, so half my students would be doing sort of applied research, and the other half would be doing fundamental research. So the applied research, what we are trying to do with the City of Calgary is trying to figure out municipal wastewater effluent is a big problem there, everywhere, all over the world, because all the treatment facilities now cannot remove all the chemicals.
Right.
And all the drugs we take, and everything goes into the water, into the sewer, and then into the water. So what we are trying to find out, what the city is interested to see is if that's going to have an impact on the biodiversity, aquatic biodiversity, and the ecosystem health, right? So what we've been doing is we've been doing studies where we are trying to. The problem with municipal wastewater effluent is it's a cocktail. There's so many different chemicals, so you could have all this additive effects or interactive effects. And so what we are trying to figure out is we are looking at it from the fish's standpoint and seeing whatever is coming out into the water, is that having an impact on them? So we're looking at the growth, we're looking at the reproduction, the behavior. All those are important for a fish living in the wild. It can affect how quickly they can eat or forage, or how quickly they can be eaten by a predator. So all those things come into play. So that's what we're looking at. We're looking at the molecular level. We are trying to see if there is a chemicals coming in the water. How is that affecting certain pathways that are important for these fish to escape or these fish to grow?
So these would be the cocktails of chemicals, or you can also do experimental studies of isolated chemicals?
We can do, yeah, and I'll get to that, because we did some studies like that, but the cocktail is, there's so much, and so the big problem is when it's a mixture, you don't know what's causing what, right? So what we do is we take fish and put them in cages in these sites, so then we know what the cocktail is doing to the animal. Then so then they monitor the water, and we know all the different chemicals that's there.
And how many do you study?
Oh, there's so many. So, there's hundreds of chemicals, especially, some of them are non-detectable. It's there in the water, but they are unable to detect it. But some common ones, to give you an example, antidepressants, it's measurable, and it's in fairly high concentration. So that's something that got me interested, in one of my students interested. And he looked at a drug called venlafaxine. It's an antidepressant, and we take that when we are depressed. It's called the serotonin norepinephrine reuptake inhibitor. It's widely prescribed, and so the concentration in the water is really high. And so anyway, he wanted to know how that's affecting the fish, and so we did a lab study where we exposed fish to different concentrations of that antidepressant, and some of the effects we've seen were really dramatic, and some of these effects were passed on to generations. For example, your father, your grandparents may have been exposed, and you were never exposed to that chemical, but you still have the effect of that. So this is being passed on from generation to generation. It's called the sort of transgenerational effect, which is really
modifying
scary.
gene expression and then from maternal to
That's right. That's right. Yeah. But the problem is there is no risk assessment. You cannot do a risk assessment, right? You don't know what's causing what. Yeah. So that's going to be a big issue in the future.
I think about that with because we run environmental science programs for undergrad and master's students. And if you look at just, we've done a survey of environmental science programs across India.
Yeah.
What you're typically measuring in water is BOD, biological oxygen demand, chemical oxygen demand, maybe nitrogen, phosphorus, a few basic things.
Yeah.
And I was in the UK year and a half back at a Royal Society Indian National Science Academy meeting, and scientists from India were presenting the work, which is again largely based on this.
Yeah.
And there they seem to have a program where they are looking at 1,000 different chemicals. So they have some 1,001, I don't know why the number.
Yeah.
But that's the number of chemicals they're measuring their waterways.
Yeah.
And their waterways actually, apparently the UK has cleaned up its waterways in the sense they look clean.
Yeah.
They have good chemical and oxygen, the oxygen levels are good, nitrogen, phosphorus is low.
Yeah.
But there's so much, like you say, the toxic chemical contaminants that are there are mind-boggling.
Yeah.
And I don't know what we have in our waters.
Yeah, exactly.
So,
because in UK, that's where this first started. Because I don't know if you heard of this gender bending chemicals in the water and all that. Because in UK, what they found was all the male fish were becoming females. Yeah, and this was because they had estrogen estrogen mimics in the water. And then they found out that it was coming mostly from birth control pills. And, so all those, chemicals, all those hormones goes into the water, and fish are exposed to that. So they found that all the male fish, all of a sudden the testes started changing into ova and ovaries.
and so when we say
This was in the 80s.
when we say there's a decline in fertility worldwide, also we don't know in human fertility. We have no idea if that is because
But we don't know. Right now there are lots of, hypotheses, and there's lots of, if you go on the internet, you'll see a lot of articles, but we really don't know what's causing what.
And you're talking right now about single drug, but then there's drug-to-drug interactions which can happen at micro concentrations.
That's right, and that's the problem with these effluents, right? Because it's all a cocktail. And looking at, like you said, there's 1,000 chemicals, right? You cannot go and test every 1,000 chemicals. It's going to take ages. But now what people are trying to do is they're trying to have cell lines, and that's where zebrafish comes into play, because they are high throughput. So you can start screening quickly and seeing, whether there are
breed very fast.
Yeah, they breed in 3 months. They breed, and so you can do multi-generational work, but also you can make transgenic zebrafish where we can put specific genes in there. And they will, have colored proteins, fluorescent tagged proteins. And so we can look at, impacts of chemicals. We can get a quick screen whether certain chemicals will have certain effects.
Wow.
Yeah. So zebrafish is becoming a really interesting model for that, for high throughput screening. And it's being used now for drug discovery and all that. Yeah, so it's got nothing to do with fishery science, but it's a nice model.
Nice model. No, absolutely. And so when you publish this research, does it actually translate into policy? Has the government decided to change something?
That's an interesting question.
That's the hard part.
That's a hard part. Yeah. But I think it's right now it's starting. Like the drug discovery has been going on, and people have been using zebrafish as a model. But from an environmental standpoint, I think there's some chemicals that they have. I don't know whether don't quote me on this. I don't know about the environmental policy, whether it's been. I think in Europe they have, like vitellogenin was one of them, which was used as a marker for xenoestrogens. Because if the male fish have the receptors for estrogen, but they don't make vitellogenin. It's a female specific protein, but then you can measure that in male fish, and if you measure that in the male fish, then you're certain that there is estrogens in the water.
Because they can't make it,
Yeah, otherwise it doesn't get activated. Yeah, so that's, but I don't know if that's in they're using that as a monitoring tool now. I'm not sure about it. And similarly, there's another another molecule called cytochrome P450-1A. So anytime a PCB acts on an animal, it activates a receptor called aryl hydrocarbon receptor, and that will activate this particular gene. And the protein is the cytochrome P450, 1A. And so you can measure that as well. You can make a transgenic animal that will, that will overexpress that when aryl hydrocarbon receptor is activated. So again, it can be used as a monitoring tool.
And I mean, because I'm assuming you've been spending some of your sabbatical year at least in India, and you do come often.
Yeah.
So do we have labs like this in India capable of doing?
Oh yeah, absolutely. There are labs, and maybe they're doing, but I'm not certain about it. But certainly, yeah, there are, the facilities are there, and
Facilities are there.
molecular, absolutely.
So if you had to think of what would be then the burning questions that we have for the future in Canada, in India, in all the places, because now I think these forever chemicals are everywhere. There's not, let's say maybe 20 years ago, the chemicals you'd find in Canada and in India would be very different. But now with globalization, I think we have pretty much the same things.
Oh yeah. And to be, again, the big problem, before it was the legacy chemicals which will last forever, right?
Yeah.
But now it's these emerging chemicals which we have no idea what they are, right? Any drug, all the designer drugs made for us eventually gets into the environment one way or the other. And now the new one now is the plastics, the microplastics and the nano plastics, and that's going to be a big issue as well in the
I
future.
was reading an article that said, and I found it quite say that if you have blinds like these,
Yeah.
and in your room, apparently, I don't know, I haven't read the original study myself, but they said the study shows that people have a far increased incidence of heart attacks, because when you pull up the blinds and pull down the blinds, there are these microplastics that get into the air and then into your bloodstream and block the heart.
That's right. And also all sorts of chemicals, like even our clothes and cushions and you name it, they all have this PFOS, and and those sort of and studies are showing that they are really bad. And when you take urine samples, you can measure them, in urine samples. So, and BPA is another one, bisphenol A from plastics. And so that's prevalent. It's everywhere, and it comes down to what it's doing in our body. We don't know, or we know to a certain extent.
So what would be, if you had to, let's say, design an undergraduate or master's program that would train the environmental scientists of the future to study what we need to study in the, let's say, 5, 10, 15, 20 years globally, what would you think are the priority areas we should be hitting?
Interesting. I think climate change is going to be a big issue, and climate change is going to trickle down to so many other changes. Flooding and sediments in the water. So along with chemicals, that's another component that's coming that will be in the picture. Along with and that's going to affect your oxygen gradient and everything. And in the ocean, it's going to be pH changes. So there's lots of changes happening related to climate change. I think climate change is going to be a big issue. But the way I look at it is, the chemicals in the environment is not going to stop, and especially with aging population, and so I think some sort of screening and an environmental program that's going to look at, in 10 to 15 years, the problems are going to be this sort of what I was telling you about, this generational effects, right? And how do you develop a risk assessment program for something like that? We really don't know, because we don't know what caused this change in that we see in a human being 3 generations down the road, right? And this person may have been in a completely sterile environment. Doesn't really matter if the gene has already been triggered or mutated due to past exposure and being transferred maternally to your progeny, then it's going to continue.
So there's no real way of resilience except to just stop the output of some of these chemicals. And
that's
then,
going to be
that will still linger across generations.
And so exactly. So like I said, your parents may have been exposed to it, and you've never seen that chemical or never been near it, but yet you feel the effect because of that, or you experience the effect because of what they've been exposed to, and that's happening. And in fish models, in animal models, people have shown that repeatedly. And it's not the DNA; it's epigenetic. So it's a regulation of the DNA that's being affected and that's being passed on. Yeah. So to me, when I think 10 or 15 years down the road, I think people should start having some sort of a monitoring program, so we know, and also at the same time developing tools to remove these chemicals from the environment. And
that could be a really big one, right?
Yeah. So right now, I know I can talk for other places, but in Calgary, the University of Calgary has a facility. We interact with the city, and they have a treatment facility which is embedded within the research component of our program. It's called AQUA. It's called Advancing Canadian Wastewater Assets. And the idea is that, so engineers can design new prototypes and they can test it. And so the effluent would come through these technology that they develop, and the effluent then would go into streams, which we have there, and we can go and test those water and see how efficient it is compared to effluent that hasn't been treated.
So this would be like adsorbents or?
Yeah, they have different types of, they have
Thing that breaks down the.
oxidation processes, and they have UVs, and so the engineers can come up with any sort of technology, and we can test the water and see, if it's actually improved, if it's removed the chemicals, and then we can develop monitoring tools. For example, let's say antidepressant, right? If they can hopefully generate instrument or a treatment facility that can, if they can remove these antidepressants, then we can monitor them in the fish and tell them, the concentration has gone down, the potency has gone down. So that's so the technology is going to play a big role, and I think a lot of universities and companies are putting money into developing technology, because that's going to be a big problem in the future. This pharmaceuticals that's going into the environment.
Because like you said, there's no other way to treat. I mean, or could a pharmaceutical company better treat its effluents? I guess this kind of programs could be employed there. Upstream.
That's an interesting point. Again, I don't know if they could couple that to something else that could be used for extracting them from wastewater. That could be one way of doing it. Absolutely. But then you have to do another risk assessment with animal model to see whether that compound is having an effect. But that would be one way to do it. If there are certain ways by which you can couple them, and so that when the drug goes into the water, either it gets metabolized and becomes less toxic, or it's coupled to a molecule that can be used for extraction and then can be removed. But the research is underway. So hopefully something like that would come out in the future.
So in that sense, environmental science to me is a very interdisciplinary field, right?
Absolutely.
You need to have. So what are the different kinds of fields that you would say your work encompasses?
Oh, yeah. So it's totally interdisciplinary. I won't go as far as transdisciplinary, because actually it is, because there's a social component as well. Absolutely, there is a social component to it. Yeah, but we use ecology, modeling, molecular biology, toxicology, physiology, developmental biology, a lot of mathematics and statistics. So so it's truly interdisciplinary. So I certainly cannot do my research if I'm just relying on molecular biology.
So which again means that students, when we train them, we must train them to be comfortable at least to moving across.
Or at least have the idea of what it encompasses, and they're aware of all the different. The way I see it is, I like technology, and when there's a new technology, I try to assimilate that in my research, and I think that's the way it should be. I think because things are changing rapidly, especially in biology and biotechnology.
Exactly.
The field is moving at such a rapid pace. Yeah.
But do you do that by, when you say assimilate new tech, do you do that by learning yourself or getting people?
No, I send people to labs where they have this this technology already, and
then they get trained.
And then they get trained. Yeah, I think I passed the stage of learning things and teaching people.
No time, I'm assuming. Which is a challenge, right? Because as you go up in the administrative ladder, then you have less time.
Yeah, like a good example is my the knockout. We generate knockout models, and I had a student doing a PhD, and she was really keen to do something like that in the lab. So I sent her to NIH just for a few days, and she learned the technique and came back, and she started that in my lab. So it's amazing, right? So and I think that's where I think that flexibility and allowing students to do what they want really helps, and listening to what they want as well.
Right.
Yeah, from a mentorship standpoint.
No, wonderful. And what are your, let's say, hopes and fears for the future, if I can say? Because now you've been doing environmental science, this kind of work for 30 years?
Yeah, more? Yeah, pretty much.
Yeah,
I started,
40.
Yeah, close to 40, including my PhD.
Yeah.
But actually, from a faculty standpoint, it's over 30 years now. Yeah.
So if you're looking, let's say, to the next 20 years, what are the hopes and fears? Because climate change, environmental stress, biodiversity collapse is all around us. But there are signs of hope at the same time.
Absolutely, yeah. I think that's where the researchers can contribute, right?
Yeah.
And because development has to take place, we cannot say stop everything. That's not going to happen, and that's not realistic. So I think coming up with programs and coming up with assessment tools and having some sort of policy as to how much a system can carry, right? And that's going to become important, and those sort of things I think in the future will become really useful. So from developing environmental scientists, I think people should be aware of the environment. They should know all the, what could happen to the environment, how various factors could impact what they see, not just, one chemical or two chemicals, but all the other abiotic and biotic factors that could come into play. And this is not just to the aquatic environment, even to terrestrial environments. I think the problem is the same regardless of what environment we're looking at. Yeah, so I think I'm hopeful. Let me put it that way. I'm truly hopeful. I think with the kind of researches we are generating now, and the technology that's out there, I think I think we should be able to at least have a handle on what's going on, and then it comes down to the government to decide what, how they want to move with it.
Yeah.
Yeah.
No, that's very nice. Thank you. I think, Matt, it was a real pleasure to talk to you and to learn about all of these things, because I think sometimes we've seen a lot of people coming in here, and young people especially like to know about the journeys. Why do people get somewhere? What motivates them? What are their hopes and dreams for the future? Because they're themselves at a stage where they're trying to figure out what to do. So
Absolutely, and my advice to them would be, just don't fall into any silos. Keep it open. Keep your mind broad. And when I was growing up, it was, either medicine or engineering, right? But there's so many options now, so many options, and then just don't stick to one career path. There's so many options, but follow your passion. I think that I tell this to everybody in my lab. If you're not interested in something, don't do it. Do it because you want to do it, and you're curious, and you want to benefit from it.
and stay ever curious, I guess.
Yeah, absolutely, and that's I think that's the most important advice I would give. Be passionate about what you're doing, and go do it.
And I think your work is a wonderful example of that. So thank you very much, Matt. It was a pleasure.
Thank you so much. Pleasure being here.